a lot of regulatory and other issues. And I met our next speaker at an X Prize event a couple years ago. Dr. Jonathan Sackler-Bernstein is a fully trained cardiologist, lots of clinical experience. Later went to the one of the good guys to go to the FDA, where he was the Associate Center Director at the FDA for Devices and Radiologic Health, so really understands the ins and outs of that realm. And more recently has been the Vice President of Clinical Development and Regulatory Affairs at Neostem, which is a company that's doing a lot of work. It's actually a public company, rolling up a lot of these technologies together. So he's going to take a look at some of the pathways to taking this forward. A shorter talk. I'll also be back this afternoon to talk more about the regulatory environment, and a workshop right after this on FDA issues overall. So lucky to have Dr. Jonathan Sackner-Bernstein. Thanks a lot.
In the 1890s, urban centers, both in the US and in Europe, were facing a major crisis. The crisis related to the fact that these urban centers were all driven by horses, that were all functioning by horses as their infrastructure. And what leaders of some of these communities, especially New York and London, noticed was that when they calculated the amount of manure that was produced on a daily basis, that they predicted within 30 to 50 years, the manure would reach the third floor windows of these cities. The whole infrastructure that was set up to bring the trains that were bringing supplies, the horses and carriages that were distributing, the beginning of people not walking and being driven around by horse-drawn carriages, were leading to an ever-increasing cycle of a problem. So they decided, the people who were then at the forefront of civil engineering as it existed there in form, if not in name, to get together to figure out what the solution would be. They called a conference in the late 1890s in New York, and while they were in New York, they said, in these 10 days, we're going to come up with a solution. But unfortunately, every single proposal involved more horses. How do you get manure? 2.5 million tons of manure a day to come out of these urban centers. 2.5 million pounds, excuse me. Well, you bring horses to carry them away. Well, you have to work the horses over and over again, then what happens when the horses die on the street? You have to cart them off with more horses. So it was an ever-spiraling problem, and even before the end of the 10-day conference, they decided that it was hopeless. They canceled the conference. They went off, figuring that the next generation of leaders would have to figure it out. Now, in parallel to this conference, there were several companies that were developing the disruptive innovation that was actually their solution. In Europe and the US, cars, the automobile was starting, the answer to the manure problem. And by the early 1900s, as cars rolled out, there was no more manure problem. A disruptive technology, a disruptive innovation, one field completely disconnected from another's problem, answered the need. And that's where we are in healthcare in many ways. We have problems. We don't even know where to look. We don't know where the solutions are. We have little fiefdoms, even within the same field. We have different industries not talking to each other. We have different kinds of experts, as Alan pointed out, that don't even like to talk in a bidirectional fashion. So what do? How do we predict where that next disruptive innovation is coming from? Well, that's really hard. But the good news is that as we sit here predicting the future of stem cell therapy and regenerative medicine, it's not really that far off. And what I want to share with you today is a couple of examples over the next few minutes of how stem cells and regenerative medicine actually exist now. It's not tomorrow. It's not science fiction. It's today. So to do so, what I want to do is talk about a couple of problems that actually were addressed and are being addressed and are about to be addressed by stem cells and regenerative medicine and tissue engineering. Depending upon what label you want to use, I think you can apply them broadly to these examples. The first has to do with a clinical phenomenon that we see as cardiologists, and I think those of us as doctors know about it if we haven't seen it directly, which is sudden cardiac death, represented here by an electrocardiogram showing a patient who went from sinus rhythm to torsade de pointes. A twisting of the axis is literally what that means, but if you look at the cardiogram, you can imagine what the patient would look like as they drop dead suddenly. And this is a known complication of drugs. It's been known for decades. There are a number of drugs that are scattered litter, pulled from the market, that have induced this abnormality, first by prolonging repolarization and then that leading to sudden cardiac death. The ability to predict that in a drug has not been what we need, and so these drugs come on the market. Sometimes there's an unanticipated interaction that raises the levels of these drugs and causes people who are healthy to drop suddenly from taking an antihistamine or a stomach medicine or an antianginal for stable angina, people who are relatively well. So to deal with this, a whole level of regulatory requirements emerged, driven partly by the science, partly by the regulators, that went from the basic cellular ion channel assays called HERG assays, all the way up to clinical trials with 50 to 100 people, normal volunteers, who are exposed to the drug to see what it does in a controlled environment. So fortunately to date, no one's ever dropped dead from being part of one of these clinical trials called thorough QT trials. But it is a place that does require significant monitoring and oversight and thoughtful execution of the trial in order to figure it out. The problem is that even with those tests, there are drugs that make it to the market that still can hurt people. There are drugs that don't make it to the market because we think they might hurt people, yet those preclinical tests are not great predictors of what happens to people. So we lose a lot of opportunities, we expose people to risks, and there really is not a great model. Well, there's not a great model that people are aware of. So if we were to look at how to look at a heart, somebody might say, well, we can grow it on a test tube, on a plate in a lab and do some tests of the effect of drugs on the heart. Well, that's awfully hard to do on a large scale. So maybe what we can do instead is look at some of these myocytes that we can take out of the heart. Now we're starting to get somewhere, because as you take these small clusters of cells and put them on a 96-well plate, if you have the right mixture and the right scale of a system, you start to see the kind of behavior that actually predicts what drugs do in people. Now, this is being done today. There's literature showing that you can take iPS cells, which have been discussed by several speakers already, from human skin cells, make cardiomyocyte-like cells. And notice I didn't say cardiomyocyte because there's some debate as to exactly whether they are cardiomyocytes or not, and we can get into that into the discussion. But the key is that when you start to look at these cells, what you see is that they behave in a way that predicts what's going to happen to people, better than those single ion channel assays, better than a clinical trial of 50 to 100 people, better than any other tool. And in this video, what you can see, what you saw initially was on the left side, before giving drugs, if you were actually to measure this carefully, you would see there's a very fixed beat frequency. And then on the right side, after a drug is given that's known to cause sudden death, essentially, and that's why it still just has a number, because this one we realized before it made it to the market, that the beat frequency starts to change. And the way this starts to show up as you look at these assays is depicted here in this graph, where you can see as you move down to the toxic drugs on the bottom levels, you see that even at low dose, the rhythmic frequency of beating goes away. There's all sorts of weird stuff that happens in the beat pattern that would reflect a mechanical arrhythmia, a cardiac arrhythmia, as you start to go out to longer-term exposures. This is for a drug, an antihistamine, that made it to the market and was on the market for years with a bunch of people dropping dead who were taking it for a stuffy nose. This is a test that can detect this better than any of the currently standard assays. So we see that tissue engineering, looking at these clusters of cells as systems, there's a sweet spot. We don't have to even get into the molecular mechanisms, the ion channels or the genetics or epigenomes. Maybe it's better to have that sweet spot in between a whole organism and those cells, where that cluster actually can predict what's going to happen clinically. So another example of where problems come to be relates to the development of treatments when we're at risk of a bioweapon. We've heard about this at this conference. It's published in the lay press and the scientific literature. What would happen if somebody developed a weapon and exposed our military or exposed our population? If we were lucky enough to have something that we thought worked, we would basically have two options to develop them. One would be something called an emergency use authorization, which in a when the president declares a national emergency, basically you can just market anything for that particular emergency, basically with no tests. So you could take a vaccine for a bioweapon and just start giving it to everybody in the military, giving it to everybody in the general population who was at sufficient risk, with no insight. On the other hand, you could say, I'm going to go ahead and give it to some rodents and some non-human primates, but if you look at the history of that, not only does it take a long time, and most of us would be dead to a weaponized virus by the time we had the data, but those models don't even predict well what's going to happen in people. So an option that DARPA funded several years ago was to figure out how to engineer a human immune system ex vivo that could predict rapidly what a vaccine would do. And this is an example shown as a diagram of how the immune system works at the top, and then how 3 key parts of it can be made to work at the bottom. The concept in this is that you don't actually need to engineer an immune system that looks like an immune system. The key is the predictive accuracy. So if you can capture the function at discrete time points and then create an automated system so that you're basically taking each of those different time point situations, the physiologies at different points when different kinds of cells would actually start to be recruited in vivo, and adding them into the models, you can create a system that seems to reflect the function of the human immune system. So the question, of course, is, does it? And there's a lot of data that show that this particular system works pretty well. This is by a company called Vax Design, which was acquired by Sanofi Pasteur a couple years ago. And this is one of the pieces of data that's real nice. They took a vaccine that potentially could be used against 2 antigens, called here antigen 1 and antigen 3, exposed the engineered human immune system ex vivo, which is basically taking a couple of people, bringing them to a blood bank, doing an apheresis to collect immature cells, and then putting on top of this platform with human umbilical vein endothelial cells and some commercial collagen. So it's about people in a scalable 96-well plate. And what they found, as you see on the right, is that there was a very pronounced response to antigen 1. And they compared that to what they saw in the standard mouse model that this particular vaccine maker used, which showed that there was a pretty similar response to antigen 1 and antigen 3. Well, what happened in people when they actually gave it in testing? Only antigen 1 actually induced a response in people. Antigen 3 induced no response in the clinical trials throughout its development. Just one piece of multiple lines of evidence that show that this engineered immune system on a 96-well plate basically allows for the rapid test within a few weeks of a new vaccine with predictive accuracy unrivaled by our standard animal test models. One more approach, which is getting more towards the therapeutic angle that I'd like to discuss with you, relates to the company that I work at now, NeoStem. One of the beauties of working in the stem cell regenerative medicine space is that as you look around, one of the conclusions that I think can be drawn pretty consistently is that these cells really know what they're doing. They're a lot smarter than we are. We think that we can get into our reductionist mode and figure out exactly what the key steps are, every single mechanism, then extract that out and come up with the 1 gene or the 2 proteins that are going to make the difference. When in reality, it's a lot more complex, and these cells have figured it out. We've evolved with a whole host of systems that can potentially take care of problems we might face. Now, if the way life works out, the evolutionary pressure doesn't call on that mechanism, it basically remains in a quiescent state, but that doesn't mean it's not available to use. So let's talk about myocardial infarction. How many people have seen patients in the general population suffer a myocardial infarction in their first 25 years of life? Because from an evolutionary point of view, maybe you've seen 1 or 2, but that's not enough to be a key factor that drives how we evolve in order to assure survival of our species. So the assumption can be that, in fact, we have the repair mechanisms to deal with a myocardial infarction, but that doesn't mean that it's actually been selected biologically. So we want to look for it. So how do we look for it? Well, in the bone marrow, there are these cells that can induce angiogenesis. That's how every organ develops. Every organ needs a blood supply. That's a common theme and a commonly recognized requisite for regeneration or generation of tissue, as well as preservation of tissue. And inside the bone marrow, there are the stem cells in each of us that actually can induce and trigger that angiogenesis. And there's a subtype that actually seems to be built to respond to an organ signaling for help. So in the myocardial infarction state, as you see on the left side of this screen, as typified by an apical infarct, there's that part that infarcts right away, that purple part. Around it, that peri-infarct zone represented in that pale, that white region, is something that is a region that's going to survive for a few weeks, and it's calling out for blood vessels. It's hypercontractile, needs more metabolic supply to make up for its adjacent infarct, and it's sending out a signal that's perfect for those bone marrow cells, but the connection isn't made. So what we do is recognizing that that small infarction will become a bigger infarction over time if we do nothing, is we go into the bone marrow, we collect those CD34 cells, we select and purify, we do potency assays, purity assays, functional assays to make sure we basically have a pharmaceutical grade therapy, like a drug, and then re-administer it back into the coronary artery through a standard balloon catheter. These cells are made to migrate to the ischemic tissue. So you give them into the coronary, they go out through the vascular wall, they stimulate angiogenesis, and perfusion is restored. We're in a phase 2 trial. There's a host of literature that says these are extremely unlikely processes to hurt somebody, although I think that if you were one of our investor relations people, you'd probably say it's a strong indicator that this is going to work. We need to prove that, obviously, in the phase 2 and the phase 3 trial. So I talked at the beginning about manure. You might wonder why I wanted to talk about manure next to stem cells. And I think it's because it gets at where we are in the development. The 1900s were the era of conquering infection. By getting rid of manure, we had clean water supply. By introducing antibiotics, we were able to fight infection. Lifespan increased dramatically. Quality of life increased dramatically. High impact stuff. And so many people will look around at the landscape and the scientific literature and say, well, that was the century of infection control. Now we're at the century of regeneration. And what I'd like to do is point out that we really are faced with a set of choices. Are our focuses, are our work going to be geared towards solving this problem tomorrow, solving this problem next year, making this the century of regeneration? Or can we look at this practically and see what's out there, the tools that are available today, the tools that are being deployed, and realize that this is now going to be today's solution, maybe this year, if you're looking at something that takes a little bit longer, but really we have an obligation to make this a century of a decade of regeneration, and the tools are there to do it. Thank you.
Awesome. We're going to hold questions because he's going to be doing a workshop right after this, and then coming back.